Hi, I'm Mary McKenna from University of Maryland School of Medicine. I'd like to thank the organizers of this workshop for inviting me to participate. I'll be talking about CNS fuels. I have no conflicts of interest to declare. I've been charged to talk about CNS fuels and these specific aspects about fuel use. My outline, I'll talk about substrate use and how not all fuels are the same, why the brain needs fuels for energy, what determines substrate use, why glucose is needed by all brain cells, use in both animal and human brain, influence of other organs, particularly as it relates to the ketogenic diet, and I'll touch briefly on which CNS fuels impact seizures and what the mechanisms are. So why does the brain need fuels, or as I will call them, energy substrates? Metabolism of fuels is crucial to provide energy for all cellular processes in brain, including ATP synthesis, maintaining ionic gradients, protein synthesis, synthesis and release of neurotransmitters, neurite outgrowth, synaptogenesis, myelination, synthesis of all your membrane lipids, including specialized lipids, maintaining reduced glutathione, and maintaining redox status in brain. Most of the brain use is related to signaling. However, housekeeping functions use around 25 to 38% depending on brain region. It's important to understand that brain metabolism is compartmentalized. This means different types of cells have different enzymes and transporters. And the metabolism in neurons and astrocytes is dynamically linked through the exchange of key compounds. This is known as metabolic trafficking, and this is required because of compartmentation in brain. Fuel use changes during development. The brain transitions from high ketone use during the suckling period to reliance on glucose as the primary fuel entering adult brain. This is influenced by the level of transporters on the blood-brain barrier and the activity of enzymes for ketone metabolism, which are high during early development and then decrease, and for glucose metabolism, which increase with maturity. Within the brain, other fuels can be used by brain cells because compounds are always released by brain cells, including ketones, lactate, glutamate, glutamine, and fatty acids. But brain cells differ in capacity to metabolize specific substrates. So use in the brain is limited by uptake across the blood-brain barrier and transport into brain cells. Metabolic capabilities of brain cells or compartmentation, which I just mentioned, and the activity and presence of enzymes needed to metabolize the fuel or substrate. And this can change when or be altered if the brain is damaged. Use is also influenced by metabolite concentration and fluctuations. Metabolites feedback and affect enzymes, and they dynamically affect the formation of transient protein complexes, which can alter metabolic activity. Post-translational modifications of proteins, such as acetylation, can greatly affect enzyme activity, and cellular damage can also affect metabolism. Rat and human brain have the same developmental sequence, but take place on a very different time scale. Human post-conception period is approximately 40 weeks until birth, and during this time there's active neurogenesis, development of immune system, and genesis of glial cells. There's some synaptogenesis going on, which continues well into the teenage years, and you also have myelination and pruning of synapses, so you maintain only the strong synapses. Rats have a much more compressed lifespan. A rat at birth is equivalent to about a 22-week post-conception human, and a 10-day-old rat pup is equivalent to a human at birth. This slide shows neuron-astrocyte interactions and specific cell-specific metabolic pathways in brain. Here we have a presynaptic and postsynaptic neurons and an astrocyte with end feet on the synapse and on the blood-brain barrier. Substrates are taken up into brain with specific transporters, GLUT1 for glucose, and MCT transporters for ketones and lactate. Glucose is taken up into neurons via the high-capacity GLUT3 transporter and metabolized via glycolysis to pyruvate, then to acetyl-CoA, entering the TCA cycle, and can give rise to glutamate. It's also metabolized via the pentose phosphate pathway. And lactate enters at pyruvate via LDH and ketone bodies at acetyl-CoA. Neurons are always draining intermediates from the TCA cycle because they're continuously making and releasing neurotransmitters. In astrocytes, glucose is taken up, metabolized via glycolysis to pyruvate, and then primarily via pyruvate carboxylation. This pyruvate carboxylase pathway is very important. It's an anaplerotic reaction, which means that it adds net carbon to the TCA cycle in astrocytes, and this leads to de novo synthesis of glutamate. Now that carbon does not necessarily stay in astrocytes because the glutamate formed is converted to glutamine, which is returned to neurons for use for making neurotransmitter glutamate or used for energy. And I should mention that glutamate neurotransmitter released by neurons is taken up primarily by astrocytes and either used for energy or converted back to glutamine and cycled back. Astrocytes are the only cells that can make glutamine, and also they make glycogen, which is an energy store, whereas neurons make GABA, use high amounts of glutamine, and form a high amount of glutamate. This is a cartoon showing glycolysis, TCA cycle, and harvest of energy from the electron transport chain. The most important point here is that glycolysis provides relatively little energy, and most of the energy is generated from the TCA cycle. Acetyl-CoA is a compound that enters the TCA cycle after condensing with oxaloacetate, and acetyl-CoA is produced from fuels, including glucose via glycolysis that we just saw, also fatty acids, and some amino acids. The reducing equivalents from glycolysis need to be transferred into the mitochondria so the energy can be harvested and so that NAD can be reoxidized. You need that for glycolysis to continue. And this requires the malate aspartate shuttle. So the acetyl-CoA and oxaloacetate condense and are metabolized in this TCA cycle, which provides lots of energy in the form of these reduced cofactors, but also provide key precursors for amino acid, purine, pyrimidine, and sterol biosynthesis. And energy is harvested via the electron transport chain and oxidative phosphorylation. During seizures, glucose metabolism can increase or decrease. There can be hypermetabolism during seizures and hypometabolism during the interictal phase. A number of brain metabolism studies have been done in epilepsy. Clearly, the most important are clinical studies using humans in vivo, where they use either PET and EEG and other imaging techniques, or ex vivo studies where they are studying surgical tissue that's been removed from the seizure area. Animal models have been used a lot and are important. Rodent models of various types of seizures, spontaneous, kindling, drug-induced, post-TBI. But in animals, in rats, the seizures are not decreased as much by the ketogenic diet as they are in humans. Studies of intact brain are particularly important, that PET and other imaging, or C13 NMR, particularly done in vivo, or ex vivo, where you inject the live animal and extract the brain. Brain slice studies are important because you can do electrophysiology, which you cannot do on these other models. And cultured cells are less useful. However, some studies with seahorse flux analyzer, respiration and acidification can be valuable. It's important to recognize that all models have limitations, and in vivo studies and those using more intact models are the most clinically relevant. The slide is a partial table of PET studies in epilepsy patients, and you can see in the interictal phase, there's usually reduced metabolism. During the ictal phase, it can be increased and decreased. It's a complex pattern. And post-ictal, also complex pattern, increased or decreased. Oxygen consumption is usually reduced in the interictal phase and increased during the ictal phase. Now, I'd like to point out two studies of glucose metabolism during seizures. These are in vivo animal studies from the Yale group. Patel et al. Showed that pyruvate carboxylase flux was unaltered. That means no effect on astrocytes in bicuculline seizures. And they also found a substantial increase in neuronal activity and glutamine cycling during intense neuronal activation. These are human studies showing areas of hypometabolism and hypermetabolism in a single human patient's brain. And you can see hypometabolism in the top panel in various areas. And here you have hypermetabolism in the same brain in different areas. So it's very complicated what is going on in human brain at any one time. This slide also shows metabolism in human brain, fluorodeoxyglucose uptake in 9 different patients. And the important point is the higher the discharge frequency correlated with higher glucose metabolism. You can see this very clearly in the bottom panel. And of course, it's not a completely straight correlation because this person had a high metabolism. But in general, it's a correlation that you find. So animal studies of glucose metabolism in epilepsy, these are 3 studies from Ursula Sonnewald's lab. One finding decreased glutamate, GABA, aspartate, and alanine formation in all areas, and showing decreased labeling of glutamine in 2 areas of brain, meaning less metabolism in astrocytes via pyruvate carboxylase, and unchanged in hippocampus. Eloquai et al. Also found decreased pyruvate carboxylation in astrocytes, and Alvstead found decreased formation of newly synthesized glutamate, glutamine, and GABA in all areas of the brain. So not all fuels are the same. Glucose has multiple metabolic fates in brain, and it has some roles that cannot be fulfilled by other substrates. It's metabolized via glycolysis to the TCA cycle, used for energy and precursors for other compounds. It's metabolized via the pentose phosphate pathway, which is really important in providing ribose for nucleotide biosynthesis and NADPH formation for lipid biosynthesis and maintaining reduced glutathione. It's used for glycogen formation and can be metabolized via pyruvate carboxylation. We'll talk about some non-glucose fuels as they relate to epilepsy, including pyruvate, lactate, ketones, fatty acids, medium-chain triglycerides, triheptanoin, and 2-deoxyglucose. This slide shows a comparison of brain fuels and what the different fuels can be used for. Glucose is our gold standard for metabolism in adult brain. It enters via hexokinase, is metabolized via glycolysis, the pentose phosphate pathway, via the TCA cycle, can give rise to glycogen formation, can be used to regenerate NAD in the cytosol, and is metabolized via the pyruvate carboxylation pathway. Pyruvate is an end product of glycolysis, so it cannot be metabolized via the pentose phosphate pathway and does not form NAD in the cytosol, but does fulfill these other roles of glucose, TCA cycle, glycogen, metabolism via PC. Lactate is converted to pyruvate via LDH. Again, cannot be metabolized via the pentose phosphate pathway, but does fulfill these other uses of glucose. Ketones enter metabolism at acetyl-CoA, cannot be metabolized via the pentose phosphate pathway or these other pathways, but can be used for energy. And it's important to note that ketones have many cellular effects. They can affect glycolysis, activate potassium ATP channels, inhibit transport into V-GLUT2, and other activities shown on later slides. Triheptanoin enters at succinyl-CoA and can be used for energy, doesn't fulfill any of these other roles, but it is anaplerotic, not metabolized via pyruvate carboxylase, but enters the TCA cycle at succinyl-CoA. Fatty acids are metabolized to acetyl-CoA and can be used for energy. And 2-deoxyglucose is converted to 2-deoxyglucose-6-phosphate by hexokinase, but it stops there and cannot be used for any of these other functions. And when you're looking at a PET scan, what you're seeing is uptake and phosphorylation of the deoxyglucose. Fatty acids from a ketogenic diet are taken up from the circulation into the liver, and they're metabolized to ketones and exported. The liver forms ketones but can't use them, so they're exported and then they enter other tissues, including brain, to be used for energy. And some glucose can also be formed. This slide from Karen Borges's group shows proposed effects of medium-chain triglycerides and triheptanoin for energy. Medium-chain triglycerides break down to short-chain or medium-chain fatty acids, which are converted to acetyl-CoA. Triheptanoin is metabolized to heptanoate, which is a C7 fatty acid, and it is converted to propionyl-CoA and then to succinyl-CoA and enters the TCA cycle and refills that C4 intermediate pool of the TCA cycle. Musi Hedera from Sonnewald's group showed that when animals were fed triheptanoin for a long period of time, the concentration of most metabolites in brain was restored to normal levels. In terms of animal studies of ketone metabolism, I'll point out in terms of studies of ketone metabolism, I'll point out two important ones. Kunicki et al. Was the first, and they infused beta-hydroxybutyrate into anesthetized rats and found there was metabolism in both astrocytes and neurons, but preferentially glial metabolism. And this was most likely because these were anesthetized rats with pentobarbital, which greatly increases neuronal activity. Julie Pan's study in 2002 was particularly important because they infused beta-hydroxybutyrate into non-fasting adult humans. And she found that it was metabolized in both neurons and astrocytes in human brain, and the metabolism was preferentially neuronal. And there have been a number of excellent recent reviews. I'll point out three of them here on beta-hydroxybutyrate and also 2-deoxyglucose metabolism in brain and why ketones have a specific effect on seizures. In summary, brain and brain cells can use many fuels for energy. There are many factors that influence metabolism. One of the most important is compartmentation, the capability of brain cells, transport, enzyme activity, concentration of other metabolites also greatly influence metabolism. Not all fuels are the same. Some glucose is always needed for that pentose phosphate pathway because you need to have ribose formed and you need that NADPH for maintaining reduced glutathione, your most important antioxidant in brain, and for forming lipids. Substrates that result in formation of NADH in the cytosol require the malate aspartate shuttle for transferring reducing equivalents into the mitochondria and reoxidizing that cytosolic NAD needed for continued glycolysis. Anaplerotic substrates are needed to replenish TCA cycle intermediates in brain. Metabolism via pyruvate carboxylate pathway is the most important anaplerotic reaction, but heptanoate, that's C7 fatty acid, is converted to propionyl-CoA and enters the TCA cycle and is also anaplerotic. Many CNS fuels have other effects and actions that may also affect epilepsy. This is particularly true of beta-hydroxybutyrate and as shown in the table. And also metabolism of glucose changes in epileptic brain. There's no studies that I know of looking, and there may be out there though, looking at ketone metabolism in epileptic brain during a seizure, but ketones generally suppress seizures. And oxidative stress, reactive oxygen species from seizures or other types of damage can damage brain enzymes and alter metabolism. Thank you.